So now we're going to look at HIV. Right, so HIV, what does it stand for? Human Immunodeficiency Virus, right? So back in the 80s, there was a high prevalence of this condition, right? So back in the day before, this is a very, very highly prevalent condition with patients that were highly sexually active and not using contraceptives, right? So they never used any kind of protection for their intercourse that was taking place, right? So this is where before the whole no glove, no love philosophy started coming in, right? So after the HIV started coming in, now everyone's like, okay, if you want love, you got to have the glove, right? Whoever says that was kind of witty at the time. Now it's completely useless, right? Because hopefully everyone is using the contraceptives while they're engaging in any kind of sexual activity. All right. Signs and symptoms of these two common conditions was Kaposi sarcoma, right? So this is a type of soft tissue cancer that was prevalent with a lot of these patients that had HIV, a high prevalence of HIV in their bloodstream. And the second condition was pneumocystic pneumonia. So pneumocystis pneumonia, right? So this is a condition where you have some kind of fluid in your lungs due to some abnormality, infection, toxin, rupture, any kind of in your alveoli, right? So these patients experience these two very common conditions that was diagnosed for HIV. And so we're going to really look at the mechanism of how HIV works. What does it lead to? How does it people end up having AIDS, right? So HIV is this little retrovirus coming in. And so the way it works is this virus comes in to these selective cells. So the specific cell that it targets are these CD4 cells. If you remember what type of cells CD4 cells were, these were our helper T cells. Remember, so the helper T cells, right? And what was the function of these helper T cells? If you remember, so let's put some RNA in this. So retrovirus means presence of using RNA, all right? So you have these T cells. What was the function of these T cells? You have two main functions. They had a big role in the cell-mediated immunity, and then you also have it in the humoral immunity. So they played a big role in the adaptive immune system, right? Whether it's for generation of antibodies or having your specific cytotoxic T cells going in battle, right? HIV comes in, binds to this CD4 receptor. CD4 receptor comes in and binds, and now the actual gene, if we were to magnetize this cell inside here, so if we were to magnetize it here, so if we're inside the entire cell now, right, and the virus comes in with its RNA, and it uses a specific enzyme called reverse transcriptase. So reverse transcriptase is an enzyme that converts RNA, let's put it here, back to DNA. So this RNA to DNA, right, so the central dogma of molecular biology when we look at the transcription translation is DNA goes to RNA, right? So this is the opposite. This goes against the central dogma. And so now this RNA that's present in the cell coming from the HIV virus gets duplicated. Now it becomes a double-stranded RNA, and it gets inserted into the genome of the actual cell, right? In a latency genome right here, right now. And during any kind of stress response or any kind of environmental factors, this causes the further replication of the virus. So remember, viruses are obligate parasites, meaning they need some kind of host to survive. No host, no instances of survival, right? So they stay in this cell until a certain area where all the genetic information is duplicated, and whenever that virus becomes active, right, it begins the lytic cycle. It begins to divide, creating more viruses and rupturing this cell, this helper T cell, and generating a high population of more viral capsids for infection of other cells, right? And so that's just a very basic mechanism of how the HIV Works, right? And so now you have these specific cells that are targeting your helper T cells. Helper T cells is part of your adaptive immune response, and so their numbers go down. So one of another labs that patients would do when they go to the doctor is they would measure their helper T cells levels, right? So they would run a CBC, complete blood count, in order to see the actual helper T cell levels. Like, what happened to it? Is it high? Is it low? Having low helper T cells can eventually lead to a condition called AIDS. And so AIDS stands for Acquired Immunodeficiency Syndrome, right? And so a lot of these patients experience AIDS, right? If the actual treatment is not created, right? So certain treatments where they block the specific enzyme or the actual binding of the virus to the cell have been used in certain drug cocktails for treatment, right? But if not treated, a lot of patients experience AIDS, which was happening in the late 1980s, early and late 1980s, right? And so what happens is when people ask me, does AIDS kill you? Well, that's kind of a trick question. So AIDS itself does not kill you, right? So all AIDS does is it suppresses your immune system. What eventually leads to the patient's death is the secondary infection. So if someone, for example, that has AIDS, right, and they're around someone that has the flu or a cold, they get the flu or the cold, now they don't have an immune system to protect themselves in, right? So that flu or cold, any kind of other secondary infection, leads to the increase in mortality of these patients.